Good afternoon and welcome to Physiology Friday Live. This afternoon we talk to Dr. Bradley Elliott about the module. We recap on the first 4 weeks of teaching. We get the opportunity to ask Brad a few questions about the module. Stay tuned to find out more, guys. Guys, welcome to Physiology Friday Live. It would be really great if everyone who is currently watching us can first of all say hello in the chat. It's always good to have everyone on board. Everything that you put in the chat today we'll be able to see. Bradley, welcome to the Wells Street studio.
Oh, Mike, I'm so pleased to be here. Thanks for organising this. Guys, for those of you who don't know, this is Dr. Mike Newell. He's a physiologist on our staff as well, and a particularly amazing technological guy. And he set up all this for us today.
Yeah, I do this every week for some of my master's students, so I feel familiar, and it's great to have Brad on board, and also so that we can speak to some of the newest members of our university as well. So I thought we should have brought some beers for this, because you know Friday transitioned into the weekend, talking about a bit of physiology. I thought that would have been nice, but I am also acutely aware that potentially some senior members of staff are watching, so.
Other drinks are available.
Other drinks are available, folks. So just so you guys can kind of navigate and see how this all works. First of all, we're going to have a bit of an opportunity to look through some slides. Just a bit of a recap of maybe the first 6 weeks first 4 weeks. 3 weeks.
It's flying by.
It feels like a lifetime at the moment, let me tell you. But we'll take the opportunity to have a bit of a recap of some of the content that Brad and the other tutors on the module have actually been covering over the last few weeks. And then we're going to open it up to you guys. So the live chat is going to be a really important thing. So I think what we'll do is if we just take the opportunity to maybe just head over there now so we can kind of see what's going on in the chat. Now, we are acutely aware that there's not a huge amount of people watching right now, but if you are, please let us know. So we're going to head across there now. Excellent. So we have some stuff that is completely irrelevant coming up here, but that's fine. Let's see if we can have a quick look. We've got Esther. How do? I'm just acutely aware that it's only Brad that you can see right now. So maybe you should have a quick...
Hi, Esther. Thanks for coming in. We've also got another student just bookmarking it for later. And that's good too. Just a reminder, this is live, but we are recording it and it'll be available afterwards. So if you don't catch it live, at least any of the information will be here later on today. You want to say hello to us? Any questions? Towards the end of the show, we'll take some live questions and answers. So if you want to queue up things you want to ask, this is the place to do it. And Mike's going to be moderating that for us as well.
Oh, hang on a second. We're having a bit of a disaster. 1 second.
Also worth pointing out that not only is he our co-presenter, as Mike is also running the show live. Particularly amazing job.
I fear I'm doing a terrible job so far, but it's fine. It's because it's a new stream and we're doing our best. So here we are. I can see some other folk coming in on the chat, but what I'm going to do now is just hand it over to Dr. Bradley Elliott so he can talk a little bit about some of the slides that we're going to drink. Now, if you have a question about any of the content on the module so far, line it up in the chat because I'm going to be monitoring that whilst Brad is talking through some of the content that you've covered over those first 3 weeks. So I'm going to politely hand over to Brad so he can cover some of the content that's been covered. And please do line your questions up in the chat from this point onwards.
Fantastic. Thanks, Mike. So what I did is we've had 3 weeks now, and I decided I'd go and chat to all of the lecturers involved and say, in the first 3 weeks, what are the key take-home points for every lecture? And this isn't going to be a repeat of lectures. We're not going to go through everything again. We don't want to take up too much of your time and just say the same thing over again. But I've tried to highlight one thing from each session that would be the bullet point. That was the take-home. This is the key slide, perhaps. And for the first 2 lectures, for the first week was pretty easy because it was me. And so I was having a look at it, having a think about it, what you did with me and what you're doing now with Steve Davis and Chris Biggs and what you're going to do in the next few weeks. And I came down to this slide. What we have here is a table looking at three types of ion channels. Oh, sorry, excuse me, three types of transmembrane proteins, and two of them specifically are ion channels. And we've got our pumps and our channels. And I thought those would be probably the key point from that first lecture. And at the time, I think I pointed this out, but I think now you're getting more of an idea of how key this is, how key it is for action potential generation at the moment, for resting membrane potential. Next week for muscle contraction, the week after that for some of the exercise physiology we'll be doing. And every week you'll be coming back to some of these basics. And so that's why it was in week 1. And that's why I think this is one of the key slides from that first lecture. From our next lecture, I'll just figure out how to transition slides here. Living the dream.
Getting there.
Transition. This is a bit of a simpler one. This lecture was more about taking data and visualizing it for future for your assessment coming up, and we will be talking about that assessment in a minute. And so this is simply put what I want a line graph to look like in my assignments when you're writing them up for me in your lab reports. And so it's a nice example of a pretend figure with some data that was made up. It's got a key, it's got labels on the axes, it's got a figure caption. You can tell the groups apart, and it's just a nice, clean figure. And if you're producing figures that look like this when you write a lab report for me, easy marks in that assessment. And so that's all for there, nothing deep and meaningful. Okay, let's see if I can figure this out for myself. Oh, no, needed Mike's help, sorry. And so this is an interesting one, because instead of a picture or a figure that you've seen before, this one is completely new. But when I was talking to Steve Davis this morning from your connective tissue and epithelia lecture, we had a chat about what the deep and meaningful part of those lectures was. And he kept coming back with his example, and then he got a pen and paper out and he drew me this picture. And so I've reproduced it here. And when it comes to both connective tissues and epithelia, it may have seemed like he gave you quite a lot of information, but everything he gave you was down to this pattern. What's the structure of the cell? What's its function? What's its location or an example of that cell? And then what example tissues do you have for that? And so you can think of all cells in physiology, or a lot of physiology, as a mix of structure, function, location, and thus what's the example tissue. Like for example, an example, nicely done. An example I use often is alveolar cells. So you've got a cell that's very, very thin. How do I do this on screen? I'm looking at myself and that's not helping. Very, very thin cell. So you've got structure there whose function is to absorb gases. So it's very thin, its structure mimics its function. Its location in the lung makes sense. And so the example is alveoli. And that makes sense, I think. And if you can consider cells from that regard, both connective tissues, epithelia, and in future when we do muscle, cardiac muscle, fat, liver cells, come back to that kind of figure. And if you can distinguish structure, function, location, and you know your cell you're talking about, I think you'll have that tissue type down and that physiological concept down in that area. All right, what have we got here? Learning about transition, fantastic. Dr. Chris Biggs's lectures, you've had 2 lectures with him now. His first one was on resting membrane potential, and his take-home point for the first lecture on resting membrane potential was a tale of 2 ions. Very poetic, Dr. Biggs. Just this concept of EM, or resting membrane potential, the difference between the inside and the outside of the cell, and how ions govern that. So what I've reproduced for you here, I think most of you can recognize this picture, is just a little neuron, maybe something long and thin, with some sodium, some potassium, and you've got our channels and our sodium-potassium pump hanging out there, and you know there's a difference going on, and that governs resting membrane potential. And Dr. Biggs would have gone into detail with you about movement of these ions up and down their gradients. When it goes down its gradient with some sort of channel flow, when it goes up its gradient with an ATP pump, and that's all good. But it's all about maintaining this resting membrane potential and getting that there's a semi-permeable membrane, the cell wall, and the movement of ions around that and making that kind of artificial gradients governs our cell's potential. And that's so important for an action potential, that very simple concept. Because once you've got that gradient, then you can leverage it and use it to do some sort of function. In this case, action potential generation. And what else we got here? What's my button? I am so lucky to have someone to help me do this. And so that brings us to the last lecture that you've had with Dr. Biggs on that action potential generation. And you may think that the key point from this would be the generation of an action potential. When you get that sodium influx and you hit that threshold potential and you get that massive self-fueling influx of sodium and you get that nice little EM change like we have imaged here, action potential generation. And yeah, that's a key point. That is important. But I'm having a chat with Dr. Biggs and he tells me that's not the key point here, Brad. The key point was, in his mind, sodium channel inactivation. At the peak of that spike, there's this absolute refractory period. And in his mind, he thought that was the key thing that students struggle with most in this concept. Not the action potential generation itself. Most people get that, and it's pretty obvious. But that question about having generated the action potential, how come it doesn't keep generating and keep self-sustaining? Why does that local area of the membrane turn off again? And he wanted to really hammer home that point about sodium channel inactivation, the absolute refractory period. So you've got your action potential traveling down your membrane and depolarizing the sodium channels next to it, and then traveling down and depolarizing next to it and traveling down incredibly rapidly. But as it jumps down the membrane, the sodium channels before it are in that absolute refractory period, and so you can't have a signal traveling both ways. Otherwise, you'd have action potentials bouncing back and forth all day long. Isn't functional, because we know that action potential movement is one way down the neuron. And so that's kind of a whistle-stop tour, a highlight reel, if you will, of 6 lectures. Real key points. Important take-homes are why these might be key points for each lecture. It's not the only thing to know, of course. You've got your learning outcomes for that. But what we wanted to do was try and make the major, what we'd call a threshold concept, the major thing that was really super important for that one lecture. And in light of these points, hopefully each lecture might make a little more sense. So, yeah, if there's any questions about that, let's see you in the comments. And perhaps maybe we'll move on to what's coming up next. Welcome back, Mike.
I learned a lot about action potentials there,
Oh,
probably
fantastic.
more than I would care to admit as an exercise physiologist, but there we are. Everyone is on a learning journey, as it were. We haven't, I'll be honest, we haven't been bowled over in the chat. I was just going to throw that out there. But we do have a few questions, or one question that's been posed, and I'll be curious to know. So Minhouse has come in and said, what kind of experiments will first years do?
Okay, that's a really good question. Because physiology is a practical hands-on science, and you know that's better than I. Dr. Mike Newell here is an exercise physiologist. He messes with people in the lab all the time, scientifically speaking.
Well, yeah, God, I'm going to get arrested after this, but yeah.
So there's 2 types of practical experiments we'll be doing in this module. During some of the tutorials, we'll be having practical tutorials. The first one of these will be next week, actually, where we have a muscle tutorial. The notes for this are online at the moment. And what we're going to be doing is measuring strength in your tutorial environment. We can't move a full gym into your tutorial rooms, unfortunately, but we've got these small grip strength meters, things that measure your strength of your forearm muscles, and we'll be doing some simple experiments around the size of muscle and the ability to produce force, and what that tells us about something called sarcomeres. And that actually, I think, is a really cool, elegant demonstration of some basic physiology. We'll do a few more of those. We'll be doing respiration, breathing stuff. We're doing blood pressure labs, a new one this year. But I guess the key thing that this question's about is what's the practical we're doing.
Well, I was going to, before we move on to that, I was going to maybe get into a bit of like, so for example, the forearm grip strength. Can you give a bit of application as to like where exactly an individual might use that? Because we're at first years here. We're kind of learning the basics moving forward, but let's like try and project ahead a little bit and get people excited about these measurements. How might you use them as a professional physiologist?
Yeah, it's actually a really good question, because it does seem kind of odd, right? Like, who cares about how strong this muscle here is? Which, when we do grip strength, this is the muscles we're talking about, the ones in your forearm.
Yeah.
Sometimes you want to measure strength in an athlete, for example, and you might go to a gym and do very high-tech, very powerful measures. a lot of my work as a researcher is working with the general population, working with large groups of people, hundreds or thousands of people, and working with non-athletes, aging populations, disease populations. And what we need is a very simple measure where we can go through people very quickly, but also very accurately. And the grip strength measure using this motion and then gripping with your hand has become a real gold standard of strength testing in aging and disease populations. And that's where we use it. It's a great predictor of health and well-being and a great predictor of longevity.
Nice. This is the thing, like maybe trying to get across the fact that these real simple basic measures actually have a real fundamental sort of place and a position within physiology as a whole, whether it's from an Asian population perspective, whether it's just looking at general function as a whole, that it's pretty important that you guys get this stuff nailed at the first instance, so that you can kind of build on that when you move through your degree programs and such like. So Minaz has come in with a fairly cheeky question here. And thank you for your question, Minaz. It's really great to have that kind of engagement in the chat. What kind of exam questions will come about membrane potentials?
Oh, very good. It's worth a try, right? You got to.
Minaz, you've fair enough, you know.
You've got to try. No, I think in a way it's a fair question. Often students think we're out to trick them or we're out to really push them, and I think if you can do the basics of membrane potential, like what's the difference between, say, a permeability of a gas versus the permeability of a sodium or potassium ion? Why is it impermeable? What are examples of it being physiologically meaningful? Those are the type of things we're looking for.
Good stuff.
Yeah, absolutely.
I always laugh. I mean, sometimes we get, when I do the different streams and stuff, I get module leaders to come in, we talk about the assessments. I think I might maybe shoehorn some of that into the session just so you guys get some benefit from that. But yeah, I mean, turn up to the sessions, get involved, ask questions, do the follow-up readings. You should be magic. But we're not going to sit here and tell you what questions are going to be on the exam, because why would we do that? So, but fair enough. Good on you for trying. So we've had, I mean, again, we've got a few people watching. We've got about 8 people watching, which is fantastic. I mean, we're less than 1% of the module at the moment, which, you know, maybe we need to work on our marketing for Physiology Friday Live, but we'll get there. Got to start somewhere. But maybe let's, whilst we wait for some extra questions coming in into chat, what can folks look forward to in the coming weeks? So we've got weeks 4, 5 before we've got student engagement week. What can we look forward to?
So next week we'll be carrying on this theme of action potentials. We'll be starting to apply them to tissue function. We'll be talking a lot about muscle and cardiac muscle. And that actually leads quite logically on from what we've been doing in the neuroscience part of the module so far. Understanding action potentials leads to an understanding of muscle contraction. And so I'll come back to see you guys next week before Dr. Biggs takes over again and finishes off a bit of a series about cardiology, blood pressure regulation, blood flow, that kind of work.
Nice.
And that takes us into the guided independent study week. Is that what we're calling it these days?
I'll go with that.
Yeah, let's go with that. Which is basically, it's a week where you guys don't have scheduled classes for most modules, and instead we give you some external activities to take part in. So for some modules and some degrees, you might be visiting museums or having external speakers come to the university. For human physiology, we're scheduling a large number of practicals for that week. And so every day that week, there will be dissection practicals and exercise physiology practicals. And if you look at your timetables, you'll see they're scheduled as one day of that week. So we run the same practicals every single day, and you guys come into one of them. And you'll get some scalpels and get to cut up some stuff and look at how hearts work. And then we'll take you into exercise labs and make you work your hearts and do some measures and do some classic physiology.
Nice.
And it should be good fun. And there's probably a few things we should talk around that. One, as you might have heard, and I've mentioned before in lectures, this is assessed. And that causes students a lot of stress, of course. And
it makes people turn up.
That's true. Maybe we should assess the stream.
Indeed. 1% for every viewed minute,
like
maybe.
it.
I don't know. Who knows? But go on.
But the practical report you write up out of the practical, that's the assessed portion. But the attendance of the practical itself is not. One of the reasons for this is because we run it in that guided independent study week. It's not technically a class week, so we can't enforce you to show up, and that's fair, I think. But attendance, of course, is useful. If you can't make it, you can't make it. on the Friday night at the end of that week, all of the data from everyone's practicals is combined together and given to the entire class, along with instructions.
Super helpful.
Yeah, and that'll be their major assessment. So
Good
looking
stuff.
forward to that.
Yeah,
It's a fun week, I think. You've done it before as well.
I have contributed to this week. I have 2 sproglets these days that keep me busy on that student engagement week. But yes,
Dr. Newell's.
mini doctor, indeed. One switched on. One is maybe he's tiny. So anyway, but we have some more comments in the chat. TG, thank you for contributing a statement here. She said, you need to promote Physiology Friday more. It can be quite interesting if more people join in with questions. Thanks for doing it. I feel like we've done a half decent job of making it look decent and put some adverts together and things, but we've got to start somewhere. So thank you for the feedback, TG. Please let your peers know that the stream is on right now and they should come and tune in, because then we can get some more questions in the chat. With the assessment on the module in mind, they have a report to put together based on the data sets. Do you want to have a bit of a chat about that? And maybe what I tend to ask module leaders when I deal with the types of streams is, how can a student do well on this type of assessment? I think we'll kick off with that one.
Yeah, I think that's a great question, isn't it? And especially maybe for the level 4 student coming to university, maybe there's a bit more nerves around these things. We like to be very clear in instructions. And one of the things we're looking for is, did you follow the instructions you were given? And that sounds really silly to say out loud sometimes, but it is really the case. And so at the end of that practical week, there'll be a very relatively clear instruction set. And it'll say things like, in 1,500 words, describe the following things. And in the first 500, talk about a few specific questions. In the second 500, give specific results and specific graphs and figures. In the third 500 words, tell me what that means. And what we're doing is essentially teaching our students to write in that kind of classic lab report structure that we've started to introduce in this module, what we call the IMRAD structure: introduction, methods, results, discussion. Very classical. The guidelines are quite clear, I think. I know you haven't seen them yet, of course, but I do think they are. And then just once you've got that, just go to the basics. Have a look back at what we've told you about how to present results in this module, and what you're learning about in your other modules, like in SHIP if you're in the life sciences, or if you're a biomed student in critical skills. Have a look at the referencing guidelines we give you, and just kind of follow those things. And those are the free marks sometimes, the formatting and the little things.
The amount of marks lost on assessments in universities because instructions are not read, or the guidance. For example, the summary that Brad provided you there with regards to presenting figures is real easy wins. Like even all the way towards master's level, we're still having these kind of basic mistakes put in. Just give yourself the best opportunity that you possibly can to get those easy wins just by following the guidance that your tutors are showing you. And it really helps, If you get that right at the start of your university life, then you're setting good habits. You're going to do well on all of the assessments that require those types of skills as you move through. And there'll be lots of them.
They're all totally transferable, aren't they?
And well, lest we not forget about employability skills here.
Heaven forbid.
Heaven forbid, indeed. The context of being able to handle data, to understand responses of large populations and grouped data, whether that's stratified by gender, by age, by BMI, whatever it's going to be, if establishing those skill sets now is going to allow you to build on that, so that when you come out of your undergraduate degree, that you're in a good place to go to an employer and say, well, actually, I feel quite comfortable handling lots of different types of data, particularly around health, well-being, and also handling medical information, really. So key skills to be had. So with that in mind, we're kind of verging on this a little bit, but to build on that kind of idea, so we've thought about how students can do well, but what types of common errors would you typically see when you're going through these types of assessments, and what types of things can people maybe try and definitely avoid doing?
Okay, it's two really good questions there. One of the classics, and one of the most common things I think we see a lot, is the last minute submission, is the work that clearly started very late in the game and was submitted very late in the game, perhaps without the last minute polish and checks and the free marks we're talking about. One of the pieces of advice I often give students for any piece of coursework is, if you have a deadline of, I don't know, the Friday, shall we say, pretend in your head the deadline is the Tuesday or the Wednesday. Just in your mind, that is now the new deadline. And finish early and have it done and dusted and put it aside. And then go do something else, anything else, for a while. And then a day later come back to it. and do last minute proofreading and looking over it and thinking about it again. Because that's when you pick up the things you probably should have got the first time through.
Fresh eyes on your own work is golden, because
So useful.
You internalize your own narrative, if you like, and you become so familiar with exactly what it is that you want to say, and that transition from what's going on in your head onto the page is not always apparent to the person who reads your piece of work.
That's very true.
And as such, until you step away from it, get fresh eyes, do something different, you can't pick out those little nuances of how that sentence reads, or the fact that you've led into a new paragraph, you've opened it up well, but then it kind of deviates and takes a bit of a sidestep, which in your mind is really clear, but in somebody else's, when they're reading it sort of not privy to your innermost feelings and thoughts, that it can actually read really strangely. So fresh
kind of
eyes.
Almost borders on another one, which I wanted to make sure I mentioned, which is it's a really weird way of articulating this, and I don't know a better way of saying it, but assignments are marked, and they get your marks, and that's the point. I get that. But when you write something scientifically, be it an assignment for me or when we write scientifically professionally, we're trying to communicate information to someone. And so if you have a read of that and have a look at that and say, is this the best way of getting my message across? If we say there's 20 marks available to make me a pretty looking figure, for example, is the figure you make the clearest way of communicating that information to me? Are you just making a figure because I want to get good marks and pass the module? Fine, fair enough. But have you made something that is really effectively telling me information? Because we are essentially storytellers in science. We're telling people complicated stories. And that can be quite hard to get the hang of. So that's why that fresh eyes can really help when you look at the work you've done and said, okay, I was tasked with telling a story about cardiovascular dynamics. Exciting topic. Have I done that well? If I read this outside of myself, have I done a good job of it as I proofread it? Do people understand what's going on? I think that would be a real good piece of advice as well, things to follow.
Nice. Good stuff. We've got a few more comments coming in on the chat. I'm going to take the opportunity now, just while we have a few viewers watching, that if you are enjoying the stream, it's really helpful for us to ensure that we know that. If you can like the video, we'll do more of it. If we don't get any likes, we're not going to do it again, because it takes ages to sort this sort of stuff out. So if you like what you're seeing, please click like. Let us know in the chat. We can always have access to the chat after the fact. And also, we will likely do another stream later on in the semester, probably promote it a little bit more this time. And can you make sure that you subscribe to the channel so that you're aware of any updates moving forward? So TG has just come back to us and said here, yes, already shared in our nutrition class group. Me and one of my peers are actually watching it together from one of the LG rooms.
Hi,
fantastic.
TG. Thanks for that.
Nice. Great to have you on board, guys. And thank you for contributing to the chat. If you have any specific questions about your level 4 human physiology module, then please just get them pinged. And it'd be really interesting to get it from a nutrition perspective as well. Obviously, this is a more generic, more encompassing, all-encompassing physiology module, but it'd be really nice to get some more specific comments from you guys as nutrition students. Minaz has got a couple more questions come in. This one's a bit more about level 4 as a whole. Minaz, it would be really useful to know exactly which course program you're on, so we can be a bit more specific. But what kind of group tasks will we need to do in the first year, and are there any presentations?
Who doesn't love a presentation?
We're presenting right now. We're honing our presentation skills, but I'm not too familiar with level 4, so I'm going to hand this politely over to Brad. So
for human physiology, you don't have any presentation assignments. So Minaz, just that's good. We're okay there. It's hard to know. I'm really sorry to answer that one clearly, because like Mike, you're alluding to, there's lots of degree pathways on this module, and there's lots of different modules that each individual person does. Of all 600 of you, I think there's a good 6 or 8 individual degrees going on with individual modules. So I'd suggest, Minaz, just have a quick look at your other module handbooks and check up on that. Yeah, it's a tough one.
Yeah, no, fair. The other question Minaz has come in, and again, Minaz, thank you so much for contributing these questions, because it makes our lives a lot easier on this end. The whole point of the stream is for you guys to ask questions. So he's highlighted here, will we get any practice assignments?
Yeah, that's a brilliant question. I think that's really useful. So you will get definitely practice assignments in human physiology. For the practical report, you might not recognize them when you see them, but you will be doing them in your tutorials. And so just to maybe articulate that a bit clearer and make everyone aware of this, when you did a very simple experiment for me 2 weeks ago, sorry, and did blood pressure, doing the nice radial artery pulse, and then took that data was for you to use and make a quick figure, and you're asked to do that tutorial. That's exactly what you'll be doing in the physiology practical report, just with slightly more complicated data. Next week, when you're doing grip strength, that has real clear applications to our exercise practical as well. And so there is, in the tutorials, especially in the practical tutorials, kind of built-in formative formative tasks that lead towards that assignment. For the final exam, I think we'll probably talk about that in the next one, but the readings you're being assigned every week, there's multiple choice questions attached to those electronically, and those are a great source of questions for us when we come to write our exam, and they'll be a great source of revision for you guys. So I'd really suggest you do those readings and do those formative MCQ questions in the reading.
Yeah, there's a tendency for students to kind of maybe poo-poo, formative assessments a little bit. And in actual fact, they are the best opportunity for you to get feedback on your performance, and that can be quite intrinsic in some ways. That sometimes you just need to know where you're at. Formative assessments have 0 credit associated to them, which is win-win. If you completely mess it up, doesn't matter. But you get the feedback to sort of say, well, it seems like you've performed pretty well on this content, whereas you might have the opportunity to actually or need to invest a little bit more time on some more specific areas where you thought, well, actually, I thought I'd got that right. But so do take those opportunities. They are super helpful, and that's why, as academics, we put them in.
Yeah, absolutely. And they can alternatively be great confidence boosters.
Indeed.
If you haven't had assessments at the university level and you don't know what's coming, then having a look at those formative assignments can be almost reassuring and be like, hey, actually, I'm doing okay at this. I know what's coming up. And that can be a real nice boost going into the assessment.
For sure. So Miznaz has come back and said, thank goodness, no presentations. TG, right now we don't have any really have any questions. I got to know about the stream only this morning. Next time I'll come up with something !
Brilliant.
Can, for this session, Bradley maybe talk about his study journey? It's always hard to visualize what we will do in the future after achieving our degree.
Wow, that's a really interesting question, TG, and then maybe a really nice way to round us off today.
Well,
it's such a long story. I don't want to take too much of your time because we all have really complicated paths to get here, don't we?
Indeed.
I was a sports science student originally, TG. So when I did my first degree, I did sports science at the University of Auckland, New Zealand. Mostly, true story, because I thought it sounded like fun.
Indeed.
And I maybe even thought it sounded easier than some of the other options. With a similar background of exercise physiology, Mike, I'm sure you'll agree that it wasn't the easiest pathway possible, and we were rudely surprised by how rigorous the degree was and how theoretically tough it was. But I really, almost by accident, got into physiology because I enjoyed what I was doing so much in the physiology modules. Off the back of my degree, I ended up working as a research assistant for the lab I was in. So after I graduated, I stayed working at the university I was in as a researcher, doing 100% pure research.
Nice. The dream.
Yeah, it is what we enjoy doing. It's why we academics love our job, it's the research we get to do. Off the back of that, I was recruited to a master's program in Canada when they needed a very special skill set that my lab in New Zealand was working on. And that was working in a hospital with clinical patients. And that was my hard career switch from being an exercise physiologist. There's recognition that exercise physiologists had particular skill sets that clinical researchers didn't have and needed.
Yeah.
And that's kind of explains my mixed heritage now that I have in my life, half exercise, half applied and clinical. Did a PhD because I enjoyed research for some weird reason. And some of us do that. We commit our lives to it. And so I spent 4 years doing a PhD off the back of my master's. And that led me to a lectureship and an academic post where I get to split my life between teaching you guys, which is great fun, really enjoy it. Supervising PhD research, which is those people who do pure research every day. Doing stuff like this, which is bizarre and great fun. What we didn't know was coming when we got into this job, right? But still great fun. And then doing a little bit of own research every now and then.
Yeah, it helps, doesn't it?
It does. It keeps the life fresh and keeps us interesting.
I find people's career pathways are always fascinating for students because you sit there as a student, sort of maybe looking at how am I going to get from where I'm at now
Yep.
in 3 years' time, 7 years' time, depending on what pathway that you take. How did these people that stood in front of me, talking with confidence about these particular topics? Confidence, sometimes, we do our best, but how did they get there? And I as a student found it fascinating as well. So yeah, thank you for sharing your
I think there's a real not concern, it's the wrong word, but it makes sense in a way, a fascination with what job am I going to get out of this? Where am I going? What's the pathway? And there's a lot of very linear diagrams that you might see in early career guidance. And a lot of us didn't take linear pathways to get where we went to.
I can give an example. Yeah. So I came out of my undergrad. I also did a BSc in Sport and Exercise Science at the University of Stirling in Scotland, the heart of Scotland. And then I was working alongside my degree, so I was working as a lifeguard, and I was also working on an ice rink, picking people up after they'd fallen over. And I got paid £1 an hour extra for lifeguarding than I did for ice skating, but I preferred the ice skating, so took the hit, because sitting on a warm poolside for hours on end is no fun. Whereas ice skating for hours all day is actually really good fun. So I took it. But in that transition between, I went from my undergraduate straight into what was essentially the first year of my PhD, but they put it as an MPhil. I think it's so they could bin me off after the
when
first
they realised
year.
you were rubbish.
Yeah, when they realised I was not great, they
Somebody
could sack me off,
that, sure.
get him out. And then they could bring someone in with a thing. Thankfully, I convinced and lied my way through the first year of my degree, PhD, to be able to stay there. But that transition from undergraduate to postgraduate life, I lived in a tent in someone's garden for 3 months.
Yes, right. I've forgotten that story.
Because I basically didn't want to move back home with my mum and dad, and I didn't have enough of a deposit to put down to live in a flat, even in Scotland, which, deposits on flats are significantly cheaper than they are here. So yeah, I lived in a tent for 3 months in someone's garden. A real low point for me was when a pig that I was in a neighbouring little pen next to us broke into my tent and knocked over all of my belongings, and I wasn't very happy.
People's homes have been broken into by the porcine species.
Indeed, Nibbles, she was called. She's now at a petting zoo, which I'm very pleased about. Anyway, we've deviated, but that's
Badly.
kind of what this is all about, really. So, I think there's some piercing insight there into some of our backgrounds, is what I would say. But yeah, no, thank you for sharing that.
No, absolutely. Thank you for sharing the story about Nibbles.
Indeed. It's maybe an overshare. I'm going to throw it out there, but it's fine. A little bit of an overshare, but dedicated to the scientific effort. And as such, I feel maybe not tent stories, but I feel like each academic that you have the opportunity to speak to and find out about their career will always have these little quirks of their pathway where, like Brad said, it's not linear. It's very much up and down, and each person will carve their own way, is what I would say. So,
and I don't think many of us saw the end when we started either.
Indeed.
You know what I mean?
Yeah, that's very true.
A lot of us ended up here because we enjoyed doing it and we just kept doing it. Yeah. It wasn't a solid plan for a lot of people. But that's okay too, I think.
Well, with that in mind, I think we may call it a day. Thank you. I think this was the inaugural Physiology Friday Live. We will certainly be doing another one in a few weeks' time. To pick up on the next few weeks of teaching, have a chance to reflect that. And we will certainly make sure that we get the link out. Maybe we'll take the opportunity to consult with you guys when would be a good time to do it.
Yeah, it's a good idea. Get a bit of feedback.
Friday 4 o'clock works for us, but does it work for you is the question. So we will try and maybe get a little bit more information as to when a stream might be good, and we'll post this Brad will post this on the Blackboard site.
Yeah, fantastic. This is being recorded, so we can take this, we'll post it, and you've got the story of Nibbles the pig forevermore.
Immortalised in Physiology Friday Live. There we are. So, right, guys, thank you very much for watching, particularly for TG and for Minaz for contributing some really great questions there.
Absolutely. Brilliant,
Thank you so much for contributing. It's been a pleasure trying to answer your questions. Just as a final thing, thank you both for sharing your story. Please, more oversharing. My biggest fear is to maybe fall in love with another subject for which I may not have the right skills later in life. We all deviate in our interests
over the place.
And you know what? Having a varied background and a varied interest actually results in innovative thinking, because you think about things in a different way to everybody else. So on that positive note, we will call it a day. Thank you very much for watching. Make sure you subscribe, make sure you like this video, and we will see you on the next episode of Physiology Friday Live. Take it easy, folks. Cheers.
See you later.